Thank you very much for the invitation. It's really great to be here. It's a fun conference, and it's also a place I've been to a couple times, so it's really great to see some familiar faces. And it's also a lot of fun to be able to tell you about some work that actually started out as a hobby that I was playing with during my postdoc in Germany, which is this species Arabidopsis arenosa. And about 5 years ago, I started my own lab, and we've been working on this thing ever since. And you may recognize this part of the name, but I do want to stress it is a different species. It's an obligate outcrosser. It looks better, smells better. It has some disadvantages, I'll admit, but it's got a lot of fun biology. All right, so another thing I wanted to mention right off is that I actually forgot to put some names on my abstract title. I was a little rushed in the last couple of weeks. So my great apologies to Chris and James. They are fantastic collaborators, and they should have been on my abstract list. But so anyway, a lot of the cytology that I'm going to show in the talk actually came from James Higgins, who's actually here at this meeting, back there somewhere in the dark. So if you want to talk to him about any of this, he's here too. And he was in Chris Franklin's lab at the time, but has his own group at Leicester now. Most of what I'm going to talk about was work from Levi Yant, who's also here, sitting next to James, actually, and Jesse Hollister, who was a former postdoc in my lab and is now starting his own group at Stony Brook. So this has been a super fun project, and again, these are the main sort of people behind what I'm going to talk about today. So we've become very interested in the idea of polyploidy, of whole genome duplication. So whole genome duplication events have happened kind of throughout the tree of eukaryotic life, mostly in plants, actually. At least most modern events seem to be happening in plants. But then there are these older events also, and these are hypothesized events in orange, down at the base of some major radiations. For example, the fishes, perhaps all the vertebrates. There are more modern events in the amphibians and fish, as well as many, many plant species. So it's been implicated in radiation, adaptation, speciation, and so forth, and certainly has played a major role in genome evolution. It's also very important in crops. So many of our crops are polyploid. You almost certainly ate one today, unless you're really, really hungry right now. If you had coffee, you had a product of a tetraploid, and if you put sugar in it, or in your tea, sorry, I added this because I'm in England. Actually, tea is not generally polyploid, but there are some polyploid strains, or tetraploid strains. So if you put sugar in that, you used what is probably the champion among crop plants as far as polyploidy, is sugarcane. It has 12 copies of every chromosome, which is totally amazing to me that that even works, but somehow sugarcane manages. It's also, again, not unique to plants. There are a number of animals that are polyploids, some of them even modern polyploids. So there are some sturgeons that are actually octoploids, so you can think of that if you ever eat caviar. It's also very important in normal development. So polyploidy happens in your cells in your body, depending on your age and your alcohol consumption levels. You have anywhere from a couple of percent to maybe about 40% of your liver cells are polyploid. Most of your megakaryocytes are polyploid, and the list goes on. This is called endopolyploidy. I'm not really going to talk about it much today, but we do think it's probably two sides of the same coin and probably is relevant. It can also go wrong, and this is a major problem. So cancer cells are often polyploid, and in fact, the most aggressive and invasive cancers in humans are in fact polyploid and aneuploid because they have problems with chromosome segregation. All right, so one of the main questions that we've become interested in is how do you make a happy polyploid? And I'll say a lot more about that in a moment. But one thing that we do know from decades of cytogenetic and other kinds of research is that when you newly form a polyploid, either naturally or with chemicals, they're highly genomically unstable. So you can get lots of rearrangements, deletions, and so forth. You get lots of epigenetic changes. They have major problems with chromosome segregation and meiosis, and that's what I'm going to talk most about today. And then there's also some evidence that they have problems with the fact that their cells get bigger, so with adjusting to the scaling problems that come with cell volume increases. However, there are a number of established, perfectly stable polyploid species out there. And these have increased fertility relative to these, so these are often highly sterile or close. And they tend to have stabilized phenotypes, genomes, and so forth. So there's a process of stabilization in here about which we know very little. So there's actually not much known about how you get from this kind of messy state to this established, stable state, especially at the genetic level. So what genes are actually driving those adaptations that you need to get from here to here is largely unknown. So to start studying this question, we've been working on a model system. This is Arabidopsis arenosa. It's found throughout Central and Northern Europe as an autotetraploid. That is, it's a within-species genome duplication. All 4 chromosomes or all 4 copies of every chromosome that it has are roughly equally homologous. There is a diploid. This is a very close relative. It overlaps with the tetraploid in the Carpathian Mountains, but is otherwise found sort of further south and into the Balkans. And here's a picture of one growing on a rock outcrop that's from here, which is where I used to live in southern Germany, and found it on a climbing trip. All right, so Arabidopsis arenosa is part of a very user-friendly genus. So this is Arabidopsis thaliana down here. And these other species are about 5 to 15 million years diverged, depending on who you ask. We have another close relative, Lirata. And Lirata, like Thaliana, has a good high-quality reference genome available, which is really useful for a lot of what we do. The diploid arenosas are currently being sequenced by JGI and also by Detlef Weigel's group. It's not available yet, however, so we've been mostly relying on aligning the sequences that we've generated to the Lirata reference. But these are actually pretty closely related, a maximum of about 2 million years diverged and probably quite a bit less. So it actually works pretty well to use Lirata as a reference. The tetraploids arose from this diploid lineage at some point in the past, but unfortunately we haven't been able to successfully date this event yet. And the main reason for that is that there actually seems to be a fair bit of ongoing gene flow between the diploid and the tetraploid, which makes dating them kind of complicated. They're at least as old as the last ice age, but they may be quite a bit older. And again, that has huge error bars on it. One big advantage to working with arenosa is that it grows in very nice places. It doesn't like vacant lots like Thaliana does. So this is Strekno Hrad, which is the castle in Slovakia. That's the home of the arenosa diploids that are being sequenced. All right. So I mentioned that there's a challenge to meiosis with becoming polyploid. So I'd like to talk to you about that a little bit more because that's mostly what I'm going to talk about today. So this is just a very brief reminder of what actually happens in meiosis. So this is a pair of chromosomes. These are 2 homologs. So the pink one and the brown one each have duplicates. They've already replicated. So these are sister chromatids, and then these are homologs. And they've crossed over in these physically visible structures called chiasmata. And that's what holds them together until they separate in anaphase. However, one of the main problems that faces polyploids is that you actually increase the number of homologous chromosomes. And the main problem with this, it seems, is that you can get crossover events onto different chromosomes. So if you look at this green chromosome here, it's recombined up here with the blue one, but then down here you have a second event onto the brown one. And the brown one is associating with the pink up here, and so on. So you get this horrible mess called a multivalent. And multivalents are strongly associated with sterility in polyploids. So they're actually really nice publications that have shown that this kind of structure is the main reason that early formed polyploids are sterile, or close to sterile. And actually there have been some really nice studies where they've done selection for fertility, and then these guys go away. Over a few generations, you can get them to disappear. These are particularly problematic when the chromosomes start to try to separate at anaphase. So the entanglements that can result can be difficult to resolve, or you can get the wrong number of centromeres going to each pole, because you have the necessary tension for the spindle to know that it has to pull this thing apart, but it doesn't know that it has to go 2 and 2. So you can get 3 going to one side and 1 going to the other side, and you end up with aneuploidy. So an improper number of chromosomes is ending up in the gametes. However, when we look at established polyploids in the wild, we'll find that they basically never do this. You almost never see wild species that form multivalents with any regularity. There are a few exceptions, but they're really rare. Instead, you find these kind of diploid-like bivalent associations. So even though there are 4 roughly equally homologous chromosomes, you'll see them kind of going off nice 2 by 2 and going through meiosis that way. And going to this kind of situation is very strongly associated with the regain of fertility. This can happen in a number of ways. One of the major ones is that you can get pairing partner preferences. So if the pink one will always choose to partner up with the brown one, then there's no problem. The green one is left to go with the blue, and you have a nice, happy situation. This is very common in so-called allopolyploid species, which have a hybrid origin. So there's already some differentiation between the different chromosome sets, and you just need to sort of firm up their preferences for each other. And this seems to be the system that's happening with a gene called PH1, which was actually worked on, molecularly characterized here by Graham Moore's group at the John Innes, so that's very cool. So PH1 is actually the only cloned sort of diploidization gene that we know of. So, and again, this is in an allopolyploid, and it's causing these kind of pairing preferences so that the chromosomes will always go with the same partner. But there's another mechanism, and that's by limiting crossovers. So if you look at this image, you'll be able to see that I drew one chiasma down here, but I didn't draw a second one up here. So if you form a crossover event here, and that somehow prevents a second one from forming anywhere along the length of this chromosome, you don't have the opportunity to cross over onto the other set. And same here for the green and the blue. And this is actually very cool, because you don't actually need to change the pairing preferences at all. There are many autopolyploids. So these are, again, species that have within-species duplications that have this kind of system where there's no preference at all. The pink may just as well choose the green or the blue, but it knows to just make the one crossover and then stop. And that prevents it from forming a multivalent, even though you have no particular preference for one or the other chromosome copy. And this seems to be the class that arenosa falls into, and I'll show you the evidence for that. So as we were starting to work on this system to try to study how you adapt meiosis to polyploidy, there were a couple questions that came up. For one thing, we need to establish that autotetraploid arenosa actually is meiotically stable. It's out there in nature, it's fertile, it's reproducing, so we think it must be, but we hadn't actually got good evidence at that point. And if so, then another important question is the diploid already good at it? So it's very rare also, but there are a few cases where there are diploid genotypes that are capable of perfectly stable tetraploid meiosis when you double them. So kale is one example. But again, most things are not in that category, but we needed to make sure that arenosa was not in that kind of rare class before trying to use it to study evolution. All right, so the first thing was to look at whether it has stable meiosis. So this is a cartoon of a metaphase spread. So these chromosomes are attached to the spindle already by these black dots, which are the centromeres. And they're about to go through anaphase. So they've already actually in this picture started to pull apart a little bit. And what we're looking for is whether they form primarily bivalents or whether you start to get these multivalent associations that I was telling you about earlier. So anything below the dotted line we might consider sort of unstable, and anything above the dotted line would be considered a stable situation. And this is a metaphase spread. It's an example, but most of the tetraploid arenosas do look like this. This is from, again, James and Chris. And what you can see here is these little blue blobs, which are actually nicely discrete. These are actually bivalents. And the reason we know they're bivalents is because there are 16 of them. And that's how many chromosomes arenosa has. So they're pairing off nicely. So these are some examples of the shapes. So these ones are called, well, these ones are probably having single crossovers. These ones, I don't know, maybe one or two. But the important thing is that in none of these spreads do we ever see multivalents. We've seen like one or two occasionally, but they're extremely, extremely rare. So essentially, we can say that the established arenosa tetraploids do not form multivalents. If you're very observant, you might have noticed that there is, in fact, one univalent here. So that's generally not considered a good situation. And the reason for that actually is because this plant is missing a chromosome. So it somatically was just lost one of them. And so this guy had nothing to pair with. But that also makes the interesting added point that it cannot gum up the works of the bivalent it should have associated with. So it's just kind of on its own. So we conclude from that yes, in general, autotetraploid arenosa is generally meiotically stable. This is also coupled with very high fertility, so they have really good pollen stainability, for example, about 97%. So then we get to the other question, are the diploids capable of polyploid meiosis? And this is important if you're trying to study the adaptation of meiosis, because you have to know that there had to be something that they adapted to. To test this, we took diploid arenosa seeds. We treated them with colchicine, which is a microtubule inhibitor that can lead to polyploidy. And then we karyotyped somatic plants to identify the tetraploids. Again, this was done by James. He counted chromosomes on lots of somatic spreads and verified that, in fact, the plants were fully tetraploid. And then he did a lot of cytology on their meiosis to see what was happening. So again, just for comparison, this is a metaphase spread from one of the natural tetraploids. Again, high fertility. And I wouldn't be here telling you this talk if we had seen the same thing in the double diploids. These are a complete mess. So you see this kind of blobs of chromosomes. You don't see the nice discrete things that you see over here. And these really extensively tangled things. So we think they're making multivalents, and they're probably kind of tying those up together, not able to resolve it properly. And this is tied with very, very low fertility. So they only have about 2 to 3% fertility. So we conclude from that no, the diploid was not already genetically competent to deal with polyploid meiosis, and there likely had to be some adaptation to get you from what would have probably been this state to something that looked like this. So then the other question is, well, if they did that, then which mechanism did they use? Did they use that preferential pairing mechanism I was telling you about, or the reduced crossover number mechanism, or something else? So to begin addressing that first question, is there a pairing preference in arenosa? We used a population genetic approach. And this was kind of just because it was easy, because we had a lot of data lying around, so we did this. This is actually a chart showing simulated data. So we simulated at a given allele frequency. If you assume that pairing preferences do exist between chromosomes, then these are the different kinds of polyploid genotypes you might expect and their frequencies. So it's just plotting how many of this genotype you would get, how many of this one, and so on. This is what it looks like when you have no pairing preferences, and you can see that these peaks are starting to really bleed together. So here these two are very discrete. Here they're overlapping, and this middle one gets much, much smaller and flatter. So that's the kind of the key one that can tell us what is going on with inheritance from natural populations. And when we look at the actual arenosa data, it looks a lot more like this. And in fact, we did do some statistical tests, and it's not statistically different from this pattern. It is statistically different from this one or any intermediate model. So we've modeled a bunch of stuff in between these two extremes, and it's different from all of them except for the extreme tetrasomic model. So what that means is that there is no evidence that there are chromosome pairing preferences in arenosa. So that means that each individual chromosome can choose any partner randomly, but then it sticks with just that partner and doesn't associate with any additional chromosomes to form a multivalent. James has also counted chiasma numbers. You can do that from looking at the shape of the chromosomes on metaphase spreads. So you can tell whether they have 1 or 2. And what they found was, James and Chris did this together. What they found was that the diploids from the Carpathians have about 1.4 chiasma per chromosome. This is actually low for the genus, so what's been reported in Thaliana and Lyrata is more like 1.8, so they seem to already kind of have been partway down this path. The tetraploid, in contrast, has about 1. It's a touch above 1. It's like 1.01 or something. So they form mostly these kinds of associations with just a single chiasma. And if you remember from the model slide, that's actually sufficient to explain why they would form only bivalents at metaphase. Okay. So far, I've told you that autotetraploid arenosa does have good meiosis. The neotetraploids, in contrast, do not, so we think the autotetraploids had to evolve to get to where they are now. And it seems that the multivalent prevention in arenosa likely evolved via reducing crossover numbers, not by getting any kind of preferential pairing like the allotetraploids commonly do. So the real meat of the story for us has been what genes are important for that meiotic stability. What are actually the molecules that are driving that? And we took a genome scanning approach to do this. So I'm going to walk you through the basics of what that actually means for those of you who are not familiar. So what we do is we can take two groups that differ in some trait. In our case, we're looking at diploids versus tetraploids. And we can sequence their entire genomes, line them all up together, and look at the differences in frequencies of alleles. Because whenever there's a gene that contributes to a phenotypic difference between two groups, you would expect that there would be different alleles in those two groups if it's relevant to that phenotype. And it looks like something like this. So this is again just a cartoon, so you're going along the chromosome here, and each of these little balls is either a window or a single SNP, and then you form some kind of genome-wide distribution, pick a cutoff, and you'll see these peaks that rise above that cutoff. And these are regions that are candidate regions for containing a gene that's important for the difference between these two groups. So we did this for arenosa. We sequenced individuals from 4 different tetraploid populations. And these are actually parts of 2 different major gene pools that we know exist in the tetraploids. They have a common origin, but they diverged after they formed. And then we have diploids from the Carpathian Mountains. So the diploids in the Carpathians actually do have gene flow with Carpathian tetraploids, but there's no evidence that they have gene flow with any of the tetraploids from the Alps. So these are the populations we sequenced. We sequenced 16 tetraploids and 8 diploids, and we aligned everything to the published Larada genome. And here are a few numbers for you. So we have 46 million sites for which we had coverage in every single individual. Those are the ones we limited ourselves to for analysis. But this was actually really good at covering most of the genes. So most genes have at least some coverage in this data set. There are 5.6 million polymorphic sites, most of them rare, so they're just found in 1 or 2 individuals. There are 1.7 million sites that are shared between the diploids and the tetraploids, and only 26 fixed differences. So this would mean that all the tetraploids have one variant and all the diploids have a different one. And actually 13 of those 26 are found in just 2 genes. So 2 of the genes I'm going to tell you about in a moment are responsible for fully half of the fixed differences between these types. So then we calculated the test statistics for various tests of selection in 100 SNP windows across the genome and tested for outliers from those distributions, those genome-wide distributions. This is what one of those looks like. This is a test that looks for differences in the site frequency spectrum between 2 populations. You get a nice normal distribution for scores across the genome. Here's our 0.5% cutoff. And then we get a bunch of genes in this kind of extreme outlier tail. And don't worry about the gene names right now. They will become relevant in a second, but I'll tell you about them. This is another example. This is a test we made up that looks for regions that are highly differentiated for their level of diversity and that have generally low diversity within the tetraploids. And again, we actually see the same group of genes. So these are all the same names in the outlier tail. So even though this is calculating a different kind of signal than this one, you still see the same genes coming up as extreme outliers, which is encouraging. This is what it looks like when you actually look at one of the chromosomes. So this is the south arm of chromosome 4. Arenosa has 8 chromosomes in the haploid set. And then this is looking at the allele frequency difference between diploids and tetraploids, so 2x versus 4x. And each dot represents an average value for a particular window of 100 SNPs. And so these are scattered above and below 0. If they're below 0, it means that the diploids have the derived alleles. And if they're above 0, it means that the tetraploids have the derived alleles. So you can see mostly it just kind of twiddles around 0. But then you have these spikes here of pretty high differentiation. And these 4 are the ones that are significant on the arm of chromosome 4. And then if we zoom in on one of these guys, this is what it looks like for an actual single peak here. So now we're looking at differentiation again between the diploid and the tetraploid, but now each dot is a single polymorphism, not a window. And so that's why the numbers are actually a little bit higher here than they are here. So again, most of the polymorphisms are not strongly differentiated, but then some rise pretty high. These up here are the gene models. And you can see that there's this one region of very, very high differentiation between diploids and tetraploids. Actually, this gene is responsible for 9 of those 26 fixed differences between diploids and tetraploids. And this falls in a gene I'm going to say more about in just a moment. But the really cool thing for us has been that there's actually no doubt what the candidate gene is there. It's not like we're looking at peaks that have many, many genes in them. It really drops back to background by the time you're out at the next gene. We have 39 peaks like this across the genome, and most of them are single gene peaks like this one. Not all of them are quite as pretty as that one, I admit. So we looked at the list of 39 genes and asked if there were any gene ontology categories that were overrepresented in that list, and it turned out there was one, and that was meiosis. So this was really exciting for us because it fits with a lot of what we know about tetraploids and some of the challenges that they face early in their evolution. This is what some of these sweeps look like, just so you get an idea. This is ZIP1, which I'll say more about again in just a second. These are actually 2 duplicated genes, so we can't tell which of the 2 actually has the sweep in it, or maybe it's both. They're both significant. This is another one. This one was actually kind of fun because this was annotated as an unknown gene, but it was such a nice sweep that I thought it has to be doing something. So we did a bunch of protein blasts, and it turned out that it was homologous to a gene called PDS5 in animals, which is an absolutely critical meiosis gene. It just hasn't been identified in Arabidopsis because there's another homolog, so there's probably some redundancy there. And then again, this is the same peak I showed you on the previous page for ASY3. These genes are all unlinked in the genome, so these are, all the dashes here are showing where the actual peaks are across the arenosa genome, and these are where the meiosis genes are falling. We did start to wonder another thing. So as we were looking at these lists of genes, we were realizing that a lot of these proteins actually are known to interact directly. And so we thought, well, maybe this is a polygenic solution of some sort, that you need a bunch of these proteins to be evolved to a particular allele state. So we asked whether there might be more subtle signatures in some of the other genes. So there are about 70 known meiosis genes in Arabidopsis. So we looked at those specifically, and this distribution shows the relationship between diversity within the tetraploids and differentiation between diploids and tetraploids. All of our sort of significant genes are down here in this quadrant, which means they have excess differentiation for their level of diversity. These 2 measures are actually positively related. So we looked for guys that were down here. And what we found was that when you look at windows in other meiosis genes in the genome, there's no evidence that they're at all different from the rest of the genome. So it looks like the 8 that we found really are kind of the special players. And even though I didn't label all of these other windows that are past the line are actually in the same genes as the ones I labeled. So just didn't want it to get too messy. So there's actually no evidence that the other genes are anything but average for the genome. So it's really, these guys are special. So what do these 8 genes do? What process are they actually involved with? So if you're like me, then these kinds of stages of meiosis would have been so far back in your memory of when you took Bio 101 in college that these might just be gray blobs in your memory. But it turns out that actually all of the important action of meiosis is happening in these stages. So it's well before metaphase, which is kind of the textbook picture you think of when you think of meiosis if you're someone like me. All right, so this is not coming out perfectly on this screen, but so I'm going to show you a picture that's kind of a process through time. And what you see here are 2 replicated sister chromatids, and they're all coiled up together here. They're being held together by these protein complexes that's called an axial element. So these are helping hold together these. They're not the only things that hold together the sister chromatids by any means, but they help kind of build this linear structure. And the two major protein components of these are ASY1 and ASY3. And ASY3 is one of our strongest peaks of selection in the arenosa genome. ASY1 is definitely up there. So these two proteins are major structural components of these chromosome axes. And these kind of form a scaffold, in a way, for a lot of what's happening during the early stages of meiosis. The SPO11 complexes, so these are the guys that are actually forming the double-strand breaks on the chromosomes that will then allow crossovers to happen down the line. These actually come to the chromosomes in a big complex, and one of the members of that complex, these SPO11 associated proteins, is PRD3, which is also under strong selection in arenosa. On top of that, we have the cohesins. So these guys hold together the sister chromatids, but they're also involved in coordinating important events and crossing over. And they also help the homologs find each other during crossing over. And three major components of these complexes are also under very strong selection in arenosa. And then you start to form something called the synaptonemal complex. So this is what's actually sort of physically holding together the homologs, which are here. And then the axial elements start to be called the lateral elements. And the middle component here, this kind of rung of this ladder, is ZIP1. And ZIP1 is that tandemly duplicated protein that's also under very strong selection in arenosa. And embedded in all this are these sites that will ultimately become crossover or non-crossover events. And it turns out mutants in the majority of these proteins in Thaliana do affect crossover rates. So it kind of fits with our model that I was telling you about, that we think crossover number is affected in these plants. So together, all these proteins help coordinate pairing, synapsis, and crossover designation. So these are really critical processes in meiosis, and they all kind of work together to make it happen. We do know that they are not true mutants, so they're not knocked out. This is a lovely picture again from James of pachytene, and these proteins are, so ZIP1 is the reddish stuff here, and that's a paired bivalent. It's already synapsed. And then this yellow stuff is ASY1, and he's done this for a couple of other proteins too. So what we know from this is that the proteins are there, and they're getting to the chromosomes. They're where they're supposed to be. We don't quite know all the dynamics of it yet, whether that differs between diploids and tetraploids, but it's not like these are mutants, which is actually not surprising because most of these proteins will make plants sterile if they're mutated. I like this thing down here because this is a bivalent here, ... and this one up here. But then when you look down here where they're separate in the axes still, so this is before they've synapsed, they're actually associated with the other partner, which would mean that if this didn't get resolved, this would be a multivalent. So we think they probably do get resolved. How exactly is the million-dollar question. But that was kind of fun to see, at least for me. All right. So when you do a genome scan like this, of course, a big question is always, do the genes that you found actually do something that you can measure and study? And so we've started to address this using a genetic approach. So this is ASY1. And ASY1 is, again, a critically important component of the chromosome axes. And what you can see here from these dots is that the top dot doesn't go quite to 1. So ASY1 is not a fixed difference between diploids and tetraploids. And it turns out that there are a few tetraploid individuals that carry one copy of this thing in a diploid state. So we genotyped a bunch of tetraploid individuals. And this is just a shorthand for designating their genotype. So this guy would have 3 tetraploid alleles and 1 diploid allele of ASY1. These guys have completely normal tetraploid meiosis. There's nothing wrong with them. They have no fertility defect. So they seem to be able to carry 1 or 2 copies of ASY1 just fine. We generated this genotype by intercrossing these guys, intercrossed those for a while. Tetraploid genetics does take a long time. And we generated F2 segregating populations that have either 4 tetraploid alleles of ASY1 or 4 diploid alleles. And I'd like to point out that these diploid homozygotes have D alleles, so diploid alleles, derived from 2 different parents. So as we're making ASY1 homozygous, it's unlikely that we've made too much of anything else homozygous. It could be, of course, but it's not very likely. All right. So not terribly surprisingly, considering what we knew about arenosa, when we look at the tetraploid allele homozygotes, everything's fine. They form nice organized bivalents at metaphase. But that's actually not true when we look at the guys that are homozygous for the diploid allele. So this, I was hoping we'd see something, but this was actually a lot more dramatic than what I expected we would see. So these guys, this is metaphase again, and you're seeing this incredible tangle of chromosomes. They're just not figuring it out at all. So these guys are tangled, they're forming multivalent associations. And when you look later, in anaphase, when these chromosomes are supposed to be nicely separated into 2 blobs, you see these things kind of strung out across the division plate. That means that single chromosomes are associating with centromeres that are going to opposite poles. And this is bad news. This means they're forming multivalents, and this probably is going to lead to aneuploidy or chromosome breakage and various other problems. Not surprisingly, these guys have very, very low fertility. They're also down around 2 to 3% like the neopolyploids are. In fact, this phenotype is very similar to what the neopolyploids do. So this was really encouraging to us because it suggested that one of the genes that we found, at least one of the genes that we found in this genome scan, really is critical for tetraploid meiosis, and its allele state does matter for the tetraploids. We're doing the same process now for a couple of the other genes, but unfortunately we don't have the results yet. It was also actually rather interesting because I came across a couple of papers pretty much right after we got this result. There's actually evidence that that many human cancers express meiosis genes. It's not completely clear why they do this, but it's polyploid cancers that start to express meiosis genes. And one of them is a homolog of ASY1, which is called HORMA-D1 in humans. And it's actually been a therapeutic target in cancers. So when you knock it out from cancer cell lines, they become less aneuploid and they become less aggressive and less invasive. So this was really curious because it suggests that having this protein there in the tetraploid context is actually bad in the context of human cancers. So how that relates to the plant situation, we don't really know yet, but it's certainly intriguing that there may be some links there. Incidentally, one of the other proteins that we find in arenosa, the homolog, is also overexpressed in human cancers. All right, so to summarize what I've told you today, we know that autotetraploid arenosa evolved stable meiosis, likely via reduced crossover numbers. And we've seen very strong signatures of selective sweeps in a number of really interesting genes that could well be involved in causing that phenotype of reduced crossing over. And we've also shown that the allele state of at least one of these genes is in fact important for tetraploid meiotic stability. So where we're going next, one of the things we're really curious about is exactly what process is failing here. Don't really know yet. This is again the ASY homozygote. How did protein functions evolve in these guys to help prevent these kinds of instabilities in the tetraploids? Another question that we've become interested in is, do these guys co-evolve? So ASY1, for example, interacts directly with ASY3, and one interesting question is going to be, if you make a change in ASY1, do you select on ASY3 or vice versa? So are these guys kind of dragging each other along as a group? We're also interested in starting to address what else might be important for adaptation to whole genome duplication in the future. I wanted to point out that Levi is starting to do some comparative genomics in other species as well to start getting at the question of whether there is, in fact, sort of a conserved system that always has to change, or whether there are multiple solutions to being polyploid. All right. So I wanted to leave you with what's probably the only haiku written about polyploids from my former postdoc, Jesse, and I'm happy to take questions.